Today I wanted to talk to you about the tobacco smoke lab. So right now I'm in the course and I'm going to show you how to get to the lab, and then we'll go ahead and get started kind of working through it and seeing what we need to do for it. So this is the course. There's two ways to get to the assignment. The first one is go to course materials, go down to content, and then click through each page of the content in the toxicology module until you find it. The other way is I have these little buttons here and they go to little newsletters, so we can find the toxicology button and we can click on it. It'll bring up the toxicology newsletter, and we can look here and we can just kind of scroll down until we find the lab. You can see here that each lab tells you where the handout is. It gives some information. If there's any questions that students have asked me, you can look here. You can see a video walkthrough that I may have for the assignment, so we can just kind of look. So here we go. Here's the lab. It's on page 5 of the module, so if you wanted to go to the content and get more information about this topic, if you went to page 5 of the module, it would have that information. We can click on the handout, see what it does. Okay, it's downloading a doc for me. Okay, so I'm going to take this document and I'm going to pull it up right next to the website, just in case I need to go on the internet, which I will. Okay, and then I'm going to kind of like work through this, and I'll put this document on the in the newsletter right here for you guys, and I'll put the video right here. That way you can use the document I have, and then if I make any notations or anything like that, that'll be there. Okay. So the first thing it says we need to do is we need to look through this tutorial. So we need to click on that link, or we can copy and paste this into our browser. So I'm going to copy and paste it into the browser and see what happens. Okay, so here's the lung toxicology, and basically you want to start here where it says begin problem set. Okay, and you're going to go through here. If there is a question, like you don't know the answer to this question, you can read the little tutorial, and you could just kind of go through and kind of learn about your lungs. And then once you've learned about the lungs, then you kind of will get into like basically what happens when things are in our lungs that we don't need or aren't very good. So here is, now we're starting to get into smoking and things that are causing death. Okay, an interesting thing that I read once is that cigarettes are the only legal product that kill half of their users, which I thought was interesting. So anyway, once we've completed the tutorial, so you'll just go through, each problem, kind of read this information. I would definitely read this information because it's good information and you'll need it for your lab. So this is kind of like when we talk about the scientific method, this part is like your background research. Okay, so that's where you'll that's what you need here. So I'm gonna make a little note here to click on the tutorial button. For information to help you answer the problem, and I'm going to highlight that. Okay, now that we've done that, we're going to click on this link, or we're going to paste this into the internet. So I'm going to take a new tab just in case I want to get back to that other one. Okay, so now we have a tobacco smoke and lung development lab or activity. Okay, it says the activity works best if you do the hypothesis, then the methods, then the data collection, then the results and interpretation. And that makes sense, right? Because that's basically, the order of a scientific method or a lab report or anything like that. Okay? So they have a hypothesis for you. If offspring are exposed to cigarette smoke while pregnant and after birth, then it will result in changes in lung growth and development of the organism. So I'll highlight that. The methods to perform the experiment are explained in the lab. Next, you will collect data. Okay, and here we've got the sections for each lab report. So I'm just going to kind of start working on these sections, and then I can copy and paste them into a new document, or I can include this document. It's up to you. Okay, so I would want, you want some sort of clear and descriptive title. I'm going to put tobacco smoke lab. That's not really like the most amazing title I've ever heard in my life, but you know, we'll go with it. For our hypothesis, we're going to use the exact same hypothesis upstairs that we had up here. So you can say my hypothesis okay. Now what I'm going to do is I'm going to go to the Dropbox folder for this assignment, and I'm going to check, because you see they don't have an introduction here. So I want to check and see if I need to write an introduction, because it said lab report, and lab reports have introductions. So let's check and see. So I'm going to go down here. Tobacco, Smoke, and Lung Development Virtual Lab. I'm going to click on that. Okay, so here's our rubric. It does require an introduction. Okay, so I'm going to add that here. I'm going to put it right here. Okay, and so what do we need in our introduction? I'm going to take this information and I'm just going to copy and paste it right here, and then I've got it right there in case I need it. And what do we have here? I'm going to put these here, and then I make sure I use I have everything I need, you know, in my lab report. I'll get rid of all these spaces and things in a minute. Then what do we need? Data collection. So basically we need data in tables, and then that lung collection data. I'm going to do it like this. There we go. Analysis and discussion. And our conclusion. Oops. Okay, and this is just going to make sure that I include in my lab report everything that I need. And that way I don't have any extra okay. There we go. So let's go back to our lab. Okay. And our introduction we can write before we've done our lab. So we need 1 to 2 informative paragraphs that describe our initial observations, a problem statement, background information, and the purpose. Okay, all of this stuff we can get from this pre-lab tutorial. That's basically the point of that pre-lab tutorial. Okay, so I'm going to highlight that, and then I'm going to make a note . So if you take some notes during that pre-lab tutorial, you know, then you'll be able to answer that part. Remember, we still have it up, so you can go and look at it. You see there's stuff about secondhand smoke. There's some more secondhand smoke stuff. Let's go back. We can talk about the number of deaths due to smoke in the United States. That would tell us, you know, why this is important, right? Right, learning about tobacco smoke is so important because, you know, it kills so many people, right? And so all of that information you can get from this tutorial. Okay, then it asks you for your hypothesis. So you know, our testable hypothesis is right there. Now when you write your introduction, you want your introduction to be in paragraph form. I've just included the bullets because I want to go ahead and I'm including the bullets because I think it's a little easier for you to like look at it and read it. But when you write these things in here, so if I was going to start writing information, you know, this lab is important because dot dot dot, you know, providing some background information, telling me about my lungs, stuff like that. All of that, you know, it would be in paragraph form. Okay, we've got our hypothesis. You can put it in your introduction and sentence form. You can write it like this. It's up to you. When you go to do your lab, I would take out this part, all of these like instructional parts. Now for your methods, you can keep methods, but I would take out this part. Okay, you want a complete listing of all your materials. Okay, so here's your introduction. They have an introduction here for you. Please don't copy this word for word. It will, the software will catch it. So now we can go to the methods. Okay, so we're using mice, we're using cigarettes, and just as you read through the lab, just kind of keep taking notes. Right, we've got, it looks like slides, and on and on and on. So just kind of keep a kind of a running list of what they're using, whatever these containers are putting the mice in, that kind of stuff. Okay, you want to use the steps followed to conduct the investigation with enough detail, but not copied directly from the website. Okay, you'll lose points for that. And then you want to list your control, your variables, any measurement techniques they used. Okay, so control group, independent, dependent, and then whatever they use to measure. Okay. It looks like they're photographing them under the microscope and doing something like that. Okay, so let's look at our data. Okay, so this is the lung tissue. This is what we're looking at, and this is how we measure septal tissue volume. Okay, basically these lines are septal tissue. Okay, so the septa are orange and the alveoli are purple. And using a technique called point counting, okay, that's a measurement technique, right? I'm going to put that there. You can estimate components of a three-dimensional structure. Okay. Now, how does this web activity differ from the actual experiment? We're going to analyze 6 samples, but you really should do 120 in the real world. Okay, the scientists would be doing this for a long time. They wouldn't just do it for a couple minutes. Okay, so here's a sample slide where the counts have been made for you. Okay, you want to type your name in here, so I'll type mine in. Okay, so here we go. We want to see where a grid crosshair touches the septal tissue. Okay, so where might we check? It looks like maybe right here would be one. Maybe right here. I'm just going to put 2 for now. There's probably more, but I'm just going to put 2 because I don't want to have the right numbers because, you know, I don't want you guys just copying my numbers. Okay, my count is too low. Make sure you try again. Okay, I'm going to put 10. Okay, my count is too low. I'm going to put 15. Your count is too high. Okay, so they'll force you to get the right number. Okay, so 12. Okay, here's the next slide. It's from an experimental group. Okay, so whatever I had there, I should have, let me skip back. Okay. So I'm going to put my data, I'm going to make a table, and then that way you guys will have this table too. What'd they say, 6? Okay, so we'll have, we'll call this group and number. Okay, so this is from the control, was the first one, and what did they say, 12? Okay, I'm going to take this and put it on 2 different pages. There we go. Okay, so this is from the experimental group. Okay, so again, we're kind of checking. I can already see that this one is less than the other one, right? Let's just put, like, I don't know, 5. Okay. Experimental. Now, I'm not going to write down the rest of these numbers here because I don't want you guys, I'm going to pause the video and get through the rest of these because I don't want you guys to see all my numbers and then not, do it yourself. So I'm going to pause the video, get through the rest of these slides, and then we'll go on from there. Okay, so I've gone through all the slides and I've got my data tables here. I'm going to blur these in the video, so don't worry about that. If you look at my paper, you'll see that I have my data table here. I have the slide number. I added that because they had it in theirs, and then I did the group, whether it was a control or experimental, and then the number of data points. So you'll want to put, do the rest. Then they'll calculate the average control for you and the average experimental, so put those numbers here. Okay, I'm going to highlight that. Okay, and so our data section says lung tissue data is included. We have that, and our data is neatly organized and presented in appropriately labeled tables or diagrams. Bam, we've got that. Our data section is done. Okay, now remember, you still have to put your data in here, but basically once you've got that, you're good to go. Okay, another thing you can do is you can just take a screenshot of this right here and put that in your data table. Okay. Now for your analysis section, it says summarize the results as an explanation of the data presented in graphs and charts. So what you could do is you could make a graph, right? I have other videos where I've showed you how to make a graph, but what I'm going to do is I'm going to we'll talk through the graph so you kind of know what you need to do for the graph. So for the graph, what I would do is I would figure out what is your independent variable and what is your dependent variable. Okay, so there's a couple ways you could do this. You could have like your, on the x-axis you could have your control and your experimental. Okay, and you could make 2 different graphs. One with control Okay, if you make this graph, you want them to be bar graphs. Okay, and then you can have your number, right, your septal number. Okay, on the y-axis. So you could write your graph like that. You could, I think that might be actually the best way to do it, because you wouldn't want slide number on the x-axis. Doesn't make a lot of sense, right? Because it's not really super useful, and slide number is just, that's random. So the 2 things you have are the control and the experimental. You could say, like, you could create a bar graph. You could have 2 bar graphs, and instead of writing control, you could have one with not exposed on the x-axis, and that's probably even better because it's actually showing you what you're doing, right? Okay. Um, and then again, you want to make sure your axes are labeled, things like that. So those are two different ways you could graph your data. Um, Again, not exposed is your control, and exposed would be experimental, just so you know what data goes with what. Okay, so hopefully this was helpful. I'm going to write in here that you can do one of these options. Okay. Or in your lab. Now that we've collected our data, we need to decide what it means. So this is our interpretation of the data. Okay, we can say that animals exposed to cigarette smoke have more septa than animals not exposed. This would give the animals exposed to smoke a larger lung surface area for gas exchange and increase lung function and compensate for damage done by cigarette smoke. Animals not exposed to ETS have less septa than animals exposed to ETS. Less septa implies that exposed animals have bigger alveoli than controls, which would lead to increased lung function. This is because bigger alveoli allow more gas exchange. And then animals exposed to cigarette smoke have less septal tissue than controls, and this means that ETS exposed animals have less surface area in their lungs and probably a reduced capacity for gas exchange. Okay, so whichever one of those you think is best, you can put those here. Okay, so you can pick the best interpretation. Okay, and that's basically the end of the lab. Okay, then we can do the rest of the conclusion. So let's see what we've got here for the rest of the conclusion. Okay, describe what our findings mean, relate them to conclusions you can draw from the data. That's basically, the interpretation. I'll put that there. Give a valid explanation based on a correct interpretation of the results in relation to the hypotheses. Again, that's your interpretation, but you want to go up and look at your hypotheses, and you want to kind of relate it to your hypotheses. So take that interpretation and relate it to your hypotheses. Take the interpretation you chose and relate it to the hypotheses. Or hypothesis. That's probably a better way to put it. And then evaluate the procedure and discuss limitation, weaknesses, any sources of error. Okay, you could talk about how maybe they cut the slides. Did they maybe not do that right? You can go back to the methods section and just look at that and see if anything, maybe there's something that maybe should be adjusted or changed, or any sort of weaknesses. Again, we only counted 6 slides, right? So are our 6 slides the best interpretation? Would our data have changed if we'd looked at 120 slides? Okay, so you want to kind of discuss that stuff. And then you want to state realistic suggestions to improve the experiment and questions for further investigation. Now this part is really important. So in real life, scientists will do an experiment. They'll make a lab report, which is basically like publishing their data. It's almost exactly like a lab report. And then at the end of their lab report or their paper, they have to discuss kind of next steps. And one of the reasons they want to discuss these next steps is because they got some sort of funding, usually a grant funding or something like that, to pay for their experiment. And so sometimes people can get additional money to continue their research, but you have to state in your paper why you need to, basically not why you need more money, but why you need to do further research, and why that's important, and what you could change about your experiment, and what how you could take it further. So that's a really important part, because it allows the scientists to kind of make their case as to why they need to keep doing the research that they're doing. Okay. And then once you've completed your lab report, you can put this in a new document, or you could just erase all this up here, and you'll write all these parts. Now, excuse me. You don't have to put this part in there, so like you would kind of cross that out when you were writing your lab report. You'll cross these things out. You won't write that. I'm not going to leave these cross outs here because I don't want you to think you don't have to do that part if you download the document. But basically that's all you have to do for this lab. So the introduction, again, you'll use that pre-lab to write that introduction. The hypothesis, your methods, and your data you'll get from this virtual experiment. And then the analysis and the conclusion section, some of that you'll get from the website, and some you won't. Okay, so hopefully this was helpful to kind of walk you through this lab. If you have any questions, please send them to me. Again, when you ask me a question, I put them anonymously, right, in the in this newsletter, and that way other people have the benefit of seeing those questions. So don't ever feel like you're wasting my time, or I bet no one else has this question. I bet you someone else does. They may not ask me, but I bet they have that question. Okay, so please, don't hesitate to ask me questions. That's what I'm here for. And I'll put them anonymously in here in a way that nobody will be able to tell that it's you. Okay, just like these are. Okay, so again, I hope that was helpful.